Plant tissue culture is a method of taking a small bit of tissue from a mother plant, putting it into a contained and controlled environment, like in a test tube on some type of gel media, and adding hormones or nutrients to create cell division in that piece of tissue and have that cell mass develop into a small plantlet. So tissue culture or micropropagation is this technique of it's basically cloning, but taking a little bit of tissue and inducing it to grow up into a full mature organism or a full plant. Hormones can be added to that little piece of tissue to make it grow roots, and also other hormones to grow stems and make it grow back into a full plant. So it's similar to taking cuttings, root cuttings or stem cuttings, leaf cuttings, and similar to cloning plants just in general, but another method where you take even a smaller piece of tissue, and it's done in sterile conditions too, so it has to be done more in like a laboratory situation for it to work. But it's becoming more and more the very common way for nurseries to increase their stock. So a lot of nurseries here around Santa Barbara, when I started working at Santa Barbara City College about 13 years ago, a lot of the nurseries got probably 10% of their plants, the baby plants, from a tissue culture facility. Places that would create hundreds of clones quickly in these laboratory situations by taking little bits of tissue and putting them on media and inducing them to grow into full-sized seedlings. So not full-size plants, but just small seedlings, mature enough to be able to be shipped to the nursery. And then the nurseries would pot them up and grow them to a bigger size where they would sell them. These days, more currently, you're seeing 30 to 40%, sometimes even more, of the stock of a nursery is purchased first from a tissue culture facility and then grown up at the nursery. So more and more tissue culture is becoming the way plants are propagated and cloned. When tissue culture is used to propagate plants, little small sections of tissue, very small, are put into these on media. This media is like a jelly-like consistency. It's made of agar, and it can be infused with nutrients and hormones and other chemicals that can promote certain types of growth in the plant. The pictures you're seeing, like on the left, is a little stem of a plant put into the media in a test tube under sterile conditions, so we don't get rotting and different pathogens that get into those test tubes. But they're done in a lab situation, sterile situation, and as the little plantlets is exposed to those different hormones, for example, IBA might be infused into the picture here, where it starts to grow roots, and then after roots, shoots, stems, and they form into a small plantlet that can then be it can be taken out of the agar, the media, and put into a regular pot in a greenhouse, out of the test tube, and become a real small seedling of a plant. And each plantlet can be subcultured to produce 4 to 6 more plants, so you can really ramp up the numbers that can be cloned this way much more quickly than with full stem cuttings like we've done so far. A little piece of a plant that's cut off the mother plant is called an explant, like excised or cut off piece of plant. You can see at the top middle there, a little bit is taken off, put onto the culture media, and then from that it grows, forming a few new stems, which all can be put into their own separate media containers, and on, so you quickly can exponentially increase the number of clones from just one small piece of tissue. All done under sterile conditions. There's I've done a fair amount of this, and there's a piece of equipment in a lab. It's called a laminar flow hood. So it's like you're sitting at a desk, but there's side walls and a hood over it. And air is blowing out from the back of this bench towards you. So it's filtered air, it's completely sterile, so wherever you work inside that little hood is sterile. There are no bacteria, spores of things floating around, viruses, they're all filtered out, and it's completely sterile back there. And that's where you would take these explants, put them into the sterile agar tubes, culture media, and so on. So you're never introducing pathogens into those sealed, sterile tubes with a little bit of explant. Because with this little teeny bit of tissue, it would rot just like that. You know, it would be very easy to get a big problem in those, so you have to be very careful. After they grow up a little bit on the agar, they form into small plantlets. You can see the little baby plantlet of this Gerber daisy in letter A. And as they grow up, they get a little bit bigger. In B, they're shipped off in these small containers. And then perhaps the nursery or even the propagator, the tissue culture lab themselves, can start separating them out and putting them into bigger pots, and they develop into, you know, real small plants like letter C, and then fully sized mature plants, letter D. This is a fully mature Gerber daisy before it starts to go to flower. This is an image from Everbloom. It's a cut flower growing nursery in Carpinteria, California, owned by the same Dutch family for over 75 years. And these are fully blooming, fully mature Gerber daisies. Up until a few years ago, the Everbloom would grow up to 275 different varieties, different colors and color combinations of these Gerber daisies, and ship them all over the Western Hemisphere. The market was becoming more and more competitive a few years ago, and their profits were declining until the point where they could barely keep afloat, and they've now switched over to 100% cannabis. The same family owns it, the same family runs it, but they've also brought in cannabis growing experts, and they're doing quite well now. So as I showed you in previous slides, hundreds of thousands of new plants and baby seedlings could be developed from just one node with this tissue culture technique. When I did this technique in college, we took a single hole puncher and would punch little circular holes in the leaves of plants and put those on the agar. So you could get 100 of them from one leaf. It's a very efficient way of cloning. It takes a little longer. With a stem, once it gets rooted, it's, you know, it's like a sizable plant. If you've got a 6-inch stem cutting and you root it and it grows leaves, you know, after a few months, it's kind of like a normal plant size. You could plant it in the ground or sell it. These tissue culture types of plants take a little bit longer, but again, you can form so many more, so much more quickly and relatively inexpensively. The reason it's not super inexpensive is because you need a lab situation. You need those laminar flow hoods. You need people to work under sterile conditions. It takes a little bit more labor, but obviously the profits outweigh the costs on this, and it has become one of the cheapest ways for nurseries to obtain plants. And tissue culture, also known as micropropagation, is done for almost all of the fruit and vegetable crops. Rarely do you see crop fields planted by seed anymore. I think corn is still done that way, but some of these tissue culture facilities are now providing farmers, like tomatoes and strawberries and things, they're being provided to farmers after they've already sprouted and purchased from a tissue culture facility. Some of the advantages of micropropagation: it's economical in time and space. Hundreds and hundreds of little plantlets in a small area can be developed. You get a greater output. You can produce millions of uniformly flowering and yielding plant clones. African Biotechnologies company produces fruit crops and bananas and indoor plant flowers. They can produce 6 million pieces, means 6 million small clones per year, for an example. These are disease-free. They're raised in sterile conditions. And the plants are usually very high quality. Tissue culture also facilitates safer movement of germplasm across nations, so you can exchange material internationally more easily because they're small and they're sterile. It's not like moving plants from one state to another, which, you know, we're always worried about moving pathogens around. These can be done when they're still in a test tube and still very small. The name of the process of taking that small piece of plant and turning it into a whole new plant is called regeneration. Here you're seeing a top-down look at a petri dish where little bits of tissue of an African violet leaf have been put on that media, and they're now sprouting new shoots, and they'll turn into quite a few, looks like dozens of new small plantlets. That's regeneration. Regeneration is possible because the plant cells that are put on that media can be made totipotent using hormones. And totipotent means the cells become reverted to a stage where they do not have yet a specific role. They're undifferentiated. Those cells on that auger can become any different type of cell in a plant. They can become a stem cell, a root cell, a leaf cell, flower cell. So it's like stem cells when we talk about mammals and humans, stem cell research. It refers to the same thing in plants, which are called meristem cells. And those are cells that are so young in their development that they have not differentiated into a certain role. Think about cells inside a growing embryo of a human. When it's really, really little, it's just a sperm and an egg unite, and it turns into 2 cells, and then 4, and then 8, and then 16. And pretty soon those cells are still just early embryonic cells. They do not have specific roles or functions, but pretty soon, as that little embryo develops, you'll get bone cells start to develop. Those cells, some of them turn into bone, some of them turn into muscle, some of them turn into vascular tissue. That's differentiation. In a plant, the meristem cells are produced by the cambium, and when they're first developed, they're totipotent, meaning totally potent. Here's another way of thinking of it, means they are totally able to be anything. They have a lot of potential, which has not yet become specified yet. But over time, as that little embryo of a plant develops, some of the cells turn into stem cells, some turn into leaf cells, some turn into root cells. So that's how regeneration happens. It's kind of like the Frankenstein process with plants. Plant hormones are naturally occurring chemicals that influence plant growth, just like in mammals and animals. Hormones induce certain changes and influence mammalian and animal cell growth and tissue growth. Growth regulators are synthetic versions of the hormones. So humans have made synthesized hormones, but they're called growth regulators. And the plant hormones are the ones that naturally occur in the plant. Cytokinins is a type of plant hormone, and it induces the production of shoots. Auxins are another type of hormone, and they induce the production of roots. Auxins are a large class of chemicals, compounds, and in which IBA, or indole butyric acid, which we've used as our rooting powder, they fall under. So IBA is an auxin. The action of the different hormones and regulators is not consistent. Different plants respond to the same chemical differently, and different plant parts from the same plant can respond differently. So it matters, and you need to do your homework to figure out which hormones, if you're going to use a growth regulator, which ones to use in which situations. Here are the steps involved in tissue culture in general. You have to start with all of the tissue you're taking from the mother plant being sterile. It has to remain sterile, free of any kind of microorganisms or pathogens and things like that. You're using sterile agar that you're putting it into. If you're putting it on a petri dish, you're using sterile petri dishes. You have gloves on, a mask on, a hairnet on, a lab coat, and you're working under a laminar flow hood. And I have done many of these types of clones, and it's very repetitive, but it's very interesting too, and you have to be very careful to make sure everything remains sterile. Also, after that, you would put them in some kind of tissue culture petri dish, and that is where the piece of tissue you took is called an explant, and you're putting it onto another dish. And at that point, the cells that you took from the mother tissue are differentiated. You could have taken tissue from a root or a leaf or a stem. So at this point, the explant has differentiated cells and cell tissues. The explants, as I said in the last slide, are plated or put onto a sterile petri dish that contains hormones and nutrients that promote the explant cells to develop into what we call a callus. It's a mass of undifferentiated cells, kind of like a tumor. So they go from being differentiated, they put on this, taken off the mother plant and put onto this into this sterile artificial environment where the cells start to proliferate, but they become these undifferentiated totipotent callus cells. Next, those individual cells of the callus are transferred sterilely or aseptically to a different petri dish containing sterile media that encourages the undifferentiated cells to become roots and shoots. So now took differentiated tissue from the mother plant, created a callus, mass of cells by growing it on a petri dish of undifferentiated cells, and then they're put onto certain types of media to make them form differentiated tissue, roots and shoots. And one mass of callus cells can be divided many times and transferred to many plates or test tubes for development of hundreds or more roots and shoots. This is the process of cloning. Once the little plants have grown up enough on their separate petri dishes and grown roots and shoots, they're transferred to soil and they grow to maturity. And at that point once they're put into a small pot and they grow a little bit more, they're ready for sale and to be shipped off to the nursery, unless the nursery is doing it themselves. The outcomes of tissue culture are plants developing from existing buds are true clones of the mother plant, just like any other type of asexual reproduction, like stem cuttings, etc. And meristematic buds tend to be protected from variations caused by environmental factors. So if you take meristem tissue, you're getting more of a true clone than if you're taking leaf tissue or root tissue that you might from somewhere else. So those are the best ones to use to make sure you're getting the exact genetic copy of the parent. There's little or no variation usually occurring when this method of tissue culture is used. And micropropagation using callus can produce variants of the original plant. So the callus can form, and sometimes you will not get all the exact same. They're similar, and they're almost always good enough clones, but that's where you might get variants, as opposed to when you're using meristematic buds. Non-meristematic cells are not protected from the variation. That's why you get changes in genetics and when those are used for cloning. When those cells, the non-meristematic cells, experience a mutation, the plants that are produced from them will show a difference from the mother plant. So saying some of the same things over again. So if you want to make sure you're getting exact duplicates, you would use meristematic tissue or buds. Non-meristematic cells will get some variation sometimes and be not quite as true to the parent. Tissue culture is also one of the methods that's used in genetic engineering. At the callus stage, a bacterium that has DNA fragments from another organism incorporates that DNA, and then that bacteria is applied, usually in like a liquid form, to the callus, and some of the callus cells will incorporate that foreign DNA and create a genetically modified organism. And the new resulting plant, in this case, would be called transgenic. But there are also other types of organisms that are transgenic, like fish, animals, yeasts, etc. The next three slides, and including this slide, are going through the methods that were used to do an explant, a tissue culture experiment in labs that we have sometimes in person. So just skim through the next, this slide and the next three, and you get a general idea of the steps that are taken if you're going to be trying to do this type of thing in a situation like a classroom or at home where you don't have a laminar flow hood and glasses and mask and all that stuff. Well, these days some of us have masks, but just you look at the basic steps that are done to do tissue culture in a home situation.